This clip shows a computer animation of a DNA molecule being copied in a living cell. A normal human cell contains 46 chromosomes, which are 23 pairs of identical chromosomes. Copy number variation are alterations of the DNA of a genome that result in a cell having a normal number of copies of one or more sections of the DNA. CNVs correspond to relatively large regions of the genome that have been deleted or duplicated. This variation accounts for roughly 12% of the human genomic DNA. For this project, we have been exploring copy number variation with data from the Genomes Project. The goal of the Genomes Project is to find most genetic variants that have frequencies of at least 1% in the population studied. As with other major human genome reference projects, data from the Genomes Project will be made available quickly to the worldwide scientific community through freely accessible public databases. This data is accessed through FTP servers that are located in the US and in Europe. In order to find copy number variations from this data, we ran a series of scripts and applications on genomic data on the FutureGrid computer India, which is located here at Indiana University. The FutureGrid project provides a capability that makes it possible for researchers to tackle complex challenges in computer science related to the use and security of grids and clouds. This testbed makes it possible for researchers to conduct experiments by submitting an experiment's plan that is then executed via a sophisticated workflow engine. The testbed includes a geographically distributed set of heterogeneous computing systems that will support virtual machine-based environments, such as OpenStack. OpenStack is a collection of open-source technology that provides scalable open-source cloud computing software. We used OpenStack Compute, or NOVA, to deploy virtual machines to run our CNV software on the 14 nodes of India running Eucalyptus cloud computing software. The methods we used to obtain CNV data from the Genomes Project started with downloading files we need from open-source servers. Then we used the Genome Analysis Toolkit, which runs a Java application to produce coverage files that are needed to run our next software, Exome CNV. Exome CNV is an R package that analyzes genome coverage files to produce copy number variations. After running this on an n number of exomes, we used a Python script that organizes the CNVs and sorts them based on mean and standard deviation of the genome's regions. These regions with high mean and relatively low standard deviations indicate hotspots in the genomic data.